Hello, everybody. Everybody who's here anyway. Where we got? We got 36 people. That's not so bad. And we have a quiz up, and this is actually going to ask you real questions this time. I hope you don't mind. It will open at 11:03, so one more minute. So we got to sit here and talk to each other for a minute. Who's got something? Claire wants to say something.
Any more woodworking drama?
Woodworking drama? Not much this week. I've been out of the basement for a while. needed a break. Maybe I'll go back down today. I've got about 3 things that are almost done. I just can't work up the attitude to do it, but maybe today I'll get them done. Okay, Farouk is here. Life is good. Ariana, Sydney, darling, I hope you're feeling better, darling. That sounded creepy. Okay, quiz is open. Go ahead and do that, and I will be back in 10 minutes exactly. Gus the dog, lays around like a big fat dog. You know it's true. I know it's harsh, but it's true. I'm not gonna lie to you. I'm not gonna lie to you. You're a sweet dog, aren't you? You're that sweet dog I keep hearing about.
Slish, you're not muted, just so you know.
I'm sorry, what's that?
You're not muted, just so you know.
Oh, yeah. Okay, thank you. I was talking to my dog. Oh, God. I'm becoming one of those crazy people who's alone too much. Okay. Okay, I think I think we're just about ready to go here. We're 11:13. Okay. So I know you saw some of these questions before, maybe all of them, but that's okay. Today's review day. Let's go over the answers. Canonical wind pathway. I don't know if you know what canonical means, but it basically means that this is the original pathway that was found for it. And this is what everyone thinks of as the wind pathway. There's another wind pathway that involves a heterotrimeric G protein, but we're not going to worry about that. So this is the one that we talked about in class. In the canonical wind pathway, the absence of the hormone Wnt phosphorylates. Oh, in the absence of hormone, GSK3 beta phosphorylates beta catenin, causing it to be ubiquitinated and degraded. When the hormone Wnt is present, it binds to frizzled, which makes a complex with LRP. This complex, excuse me, this complex attracts disheveled to the complex. It binds to axon. Oops, nope. That's not it. Okay, it binds to TCF-LF, becomes a transcription factor. See if I was right. I was right. Okay, so that's the first one. Second.
I have a question, if you don't mind.
Okay.
So like I wrote that it creates a complex with disheveled, and this complex attracts axon to the
Oh,
complex. So like,
yeah,
I mean, what do you
that's
think?
true also.
Okay.
Oh, yeah, actually, yes. LRP. Yeah, actually, either of those would be right. I'll go through and fix that. Give you guys credit.
All right. Perfect.
Good question. Okay, so you've seen this one before. A pharmacological antagonist blocks it. A pharmacological agonist activates the receptor. For example, smooth muscles contracted by calcium, relaxed by cyclic AMP. So that's physiological antagonism. Okay, that was pretty easy, I think. I have a PhD in cardiovascular pharmacology, so maybe I'm not the best judge. Okay, when a receptor,
I'm sorry. Could you go back?
sure, it would be an honor. Okay,
Yeah. Mhm. The smooth muscle is contracted by calcium, right?
yeah, did I put them in the wrong place? I don't know if I
Yeah,
even
I
read
think
it, I
so.
just threw them up there. Contracted by calcium, relaxed by cyclic AMP.
Yeah, that looks good now.
Okay, yeah, I was doing it too quickly.
And that's not saying that smooth muscle contracted by calcium and relaxed by cyclic AMP is antagonistic.
It's saying that calcium and cyclic AMP are antagonistic to each other. So that represents a physiological antagonism. One hormone causes contraction, the other causes relaxation, but you're not antagonizing at the receptor level, but at some second messenger level down below.
Okay, got it. Thank you.
When a receptor is overstimulated, it becomes desensitized or downregulated. Let's say downregulated. When this occurs, the blank subunit of the G protein, that would be the beta GAB subunit, recruits beta adrenergic receptor kinase to the membrane, which phosphorylates the receptor. This attracts beta arrestin to the receptor. This blocks the interaction with G proteins, causing desensitization, and targets it for accumulating in calatherin-coated pits. Okay, that's our receptor downregulation. Okay, everybody good with that? Any questions, comments? Okay, so let's do our review today. All I'm going to do is start off with the PowerPoint and just go through the PowerPoint and hit the highlights as we go through. Any questions, don't be afraid to stop me, okay? And ask me more in-depth question. I may not, maybe Lake, I can see your face, Lake. Can you just tell me if something comes up on the chat? Because sometimes I can't get to it. Is that cool?
Yeah.
Okay. Messengers and receptors. Okay. So the first thing
I have a question.
Okay, go ahead.
For the exam, because I know you had mentioned, like, I had it in my planner that it was subject to change. Is it for sure on Tuesday?
Yep, it's Tuesday.
You're muted. I can't hear you.
Oh, shoot.
Oh, you said Tuesday?
It's Tuesday, yeah. It's definitely Tuesday.
Thank you.
That's Rachel, isn't it?
Yes, it is.
Wasn't I supposed to do something for you on Tuesday?
Yes, you were.
And I didn't, did I?
I don't think you did.
And you didn't call me a dirty name, did you?
No, I'm actually very nice. I'm not.
Thank you. Thank you. You know I deserved it. I'm gonna write myself a note so I fix that. Sorry. See, this is what it is to get old. You're gonna ask Barbara about this. You do it right away, because if you say I'll do that later, it's gone. Okay, let's get back to this. Oops. Oh, shoot. Okay, so the first, you can see this PowerPoint, right? Autocrine, paracrine. So the first thing we talked about is kind of the generalness of how hormones are released. What I always thought of as a hormone was this, which is released by a cell in one tissue, carried by the bloodstream to another tissue, and activates any cell that has the receptors on it for it. That's endocrine. Endocrine, which means inside release, basically, released inside your body. Here is paracrine. Para is, they're right next to each other, paracrine. And like the synapses are paracrine release. And then you've got autocrine, where one cell releases something and the same cell responds to it. And the fourth type is juxtacrine, where the receptors on one cell and the ligand for the receptor, I hate to call it a hormone, but the ligand for the receptors on another cell. So the two cells come close together and one cell touches the other cell, ligand to receptor, and that activates signal transduction. We'll see that with apoptosis in the next chapter. And also we saw it in developmental biology, if you were there, with the Notch receptor. Okay, so that's types of release. Okay, now types of hormones, two different types of hormones. One is your lipophilic hormone, and this is all I say about it. Doesn't mean it's not important, just means there's not a whole hell of a lot to say about it. Basically, your hormone is lipophilic, which means it can diffuse into the cell, which means that its receptor is going to be in the cytoplasm or in the nucleus already. So that hormone then comes down, binds to its receptor, and then it becomes a transcription factor that activates a whole suite of genes that are related to that hormone. Okay, so that's your lipophilic hormone. Your hydrophilic hormone, which is most of them, have got a cell surface receptor. Because they're hydrophilic, they can't get into the cell. So they bind to the cell surface receptor, which activates signal transduction, which then causes changes in the cell. Now, we talked about two different types of changes in the cell. One are cellular responses, cyclic AMP and IP3, which physiologically antagonize each other. And these are fast, quick, rapidly developed responses that can also be reversible and go back. So it's, you know, maintaining your heart rate and your blood pressure and everything else in your body that's constantly changing, your body adapting to it. The other is when you cause changes in gene expression, and these take longer to come on, and they tend to be permanent. So here we're talking about growth factor receptors activating differentiation of the cell, and now it's become a different cell, and it's never going back to that old cell again. So that's the other type of signal. Okay, blah, blah, blah. Amplification, right? Part of what's important about this signal transduction pathway is, when we go through this, there's always these big long pathways, and it's like, why do I have to know all this? Well, the thing about a long pathway is it gives the cell the opportunity to amplify the signal, but also different places that can be modulated, so it can be fine-tuned. So longer pathways are more important for the cell. But anyway, here is an example of how things can be amplified through a signal transduction pathway. And some interactions are amplifications, like here from 10 to the second to 10 to the fourth, and some are not, 10 to the second to 10 to the second. And the way you tell the difference is if you've got a stoichiometric interaction, it's not going to be amplification. So the alpha subunit binds to adenyl cyclase, that's 1-to-1, that's not amplifying. Once AC is on, though, you make a lot of cyclic AMP. So one molecule of AC makes 100 of cyclic AMP. So that's amplification. So as we go through these first couple mechanisms, think about how the signal gets amplified through them. Here's our receptor regulation, which we talked about. I think you can go there and look at this if you want to. No, you can't. That's in Flash. Yeah, you can't look at that. But anyway, the receptor activates the G protein. The beta gamma activates GRK, which then phosphorylates the receptor and brings beta arrestin in. And that causes the whole thing to be internalized. And this part of it was about regulation. So the more you stimulate the cell with a specific hormone, the more receptors get taken away from the membrane, and they may even be degraded by the lysosome. If you have an understimulated cell, it'll put more receptors into the membrane, so you get a super sensitive cell, and it will even like make new receptors and put them up into the membrane as well. So the number of receptors at the cell surface is going to be regulated by how much stimulation it gets. If it's overstimulated, they downregulate. If it's understimulated, it upregulates. Here are the three different receptors, or three different functional domains of receptor. You've got your ligand binding site, the site that interacts with the second messenger system like a G protein, and the regulatory sites. And here is our binding site. You can see how the ligand fits perfectly into this little gap here and interacts with the side chains of the amino acids. Here is our activation domain over here, which is going to interact with the G protein, cause it to let go of GDP, and then it will pick up GTP later. And then you've got phosphorylation sites on the tail that are going to be for regulation. Here is our space filling model. Okay, G protein cycling. So G proteins, when they're off, have GDP on the alpha subunit, and the beta gamma is just sitting around there hanging out with it. When you have an activated receptor, it is a GEF, guanine nucleotide exchange factor. It causes GDP to be released and then GTP to be picked up. And then the alpha and beta separate from each other. The alpha goes off and activates some effector like cyclic AMP, or adenyl cyclase or phospholipase C, some other ones too, some channels or whatever. But the beta gamma typically isn't doing a lot. We just saw that it can activate GRK. It can also activate some potassium channels. So it does have a role sometimes, but most important thing is once GTP is around, they separate from each other. And now the alpha subunit will break down that GTP and go back into its inactive form. It's a protein called GAP that can help. With these large heterotrimeric G proteins, it really doesn't need a GTPase activating protein. The small monomerics do, but the large one really doesn't. But it still is important here as well. Okay, so that's your cycling. As long as you've got an active receptor, it'll keep cycling like this, which means you'll always have a certain concentration of alpha subunit active. As soon as there's no more hormone around, the receptor gets turned off and all the G protein goes back into this inactive state over here. And that's what's linking your receptor to the effector. And here it is in a more schematic form or whatever. Okay, so our second messenger is adenyl cyclase. Here is ATP. Here's our ribose, adenine, and 3 phosphates. This oxygen will come down and replace that oxygen and make a circle here. So that's cyclic AMP, 1 phosphate, MP. And that will then go off and activate protein kinase A. And this will constantly be broken down by phosphodiesterase to give you AMP, which will then, go away and be made into ATP again. So kind of steady state, the way you want to think about this, you always have ATP around, okay? And then when you get an activated adenyl cyclase, you start making cyclic AMP. And you always have phosphodiesterase around. So as you're making cyclic AMP, you're breaking it down. So this is kind of a cycle too. Well, not really a cycle because this doesn't go back up there directly, but this is being made and it's being broken down, being made and broken down constantly. But because it's constantly being made, its level comes up. So now it's high and it's producing an effect. As soon as that receptors goes away and the G protein turns off, it comes down because phosphodiesterase is there. Okay. Steady state, baby. And this is just that, your receptor activating a G protein, GS. Remember, S is important here because there are isoforms of that alpha subunit. Activating adenyl cyclase, making cyclic AMP. And cyclic AMP then will activate protein kinase A. So protein kinase A is 4 subunits, heterotetramer, I guess. 2 regulatory subunits and 2 catalytic subunits. When cyclic AMP is around, it binds to the regulatory subunit, and those 2 regulatory stay together, the catalytics separate, and then you've got an active adenyl cyclase. In a lab, you can isolate just the catalytic subunit and or even get the DNA for it and express it in a cell, and it's going to be constitutively active. The only way to shut this off is to have the regulatory subunit there to bind to it. Okay, so that's protein kinase A. And protein kinase A is a serine threonine kinase. It's going to phosphorylate in specific consensus sequences, RRX serine or threonine, or RR serine or threonine, and only in those sites. So this is going to turn on specific proteins, cause specific changes in the cell. Typically, we associate cyclic AMP with the fight or flight system, so it's kind of stimulatory, activating the physiology to I don't know, do stuff. Increase energy production, increase oxygen blood flow and everything like that. So here's some examples of what it does. I would never, ever, ever ask you about this on a test. Maybe if you took cardiovascular pharmacology, but not in this class. Here's our other physiological, what do you call? Physiological second messenger system, and that's PIP2. So you've got glycerol and 2 fatty acid chains linked through a phosphate to inositol. And inositol is phosphorylated on the 4 and 5 position, making this phosphotyl-inositol 4,5 bisphosphate. So we've talked about this a lot in this class. This is so important. It's used in so many different things. So this membrane phospholipid then can be cleaved by phospholipase C, cleaves it off here, and that gives you IP3 and diacylglycerol. And both of these are going to be second messengers. And here's the production of it there. So IP3 is going to activate the IP3 receptor. I think okay, this is what I like better. It's going to activate your IP3 receptor, which is similar to your ryanodine receptor, similar in structure. It's kind of an isomer of it, but this is going to respond to IP3 only. That causes the channel to open, calcium comes out, calcium increases in the cell. Also, your diacylglycerol will activate protein kinase C. Protein kinase C can do a lot of different things in the cell, but one of the things it can do is increase calcium coming in. So both of these things are increasing calcium in the cell. What's that, Lake? What does calcium do? I'm glad you asked. Calcium binds to calmodulin. And I love a protein that tells you what it does. It modulates things based on calcium concentration. So calmodulin will bind 4 ions of calcium and then change conformation. And it will then bind to target proteins and turn them on. So calmodulin is really important for regulating things in the cell. Kind of things it'll turn on are, one is the calcium calmodulin dependent protein kinase, calcium calmodulin dependent protein kinase. Again, an enzyme that tells you what it does. It also activates myosin light chain kinase, if you remember that from chapter 14. And it activates nitric oxide synthase that we'll see in a second. It's going to make NO. And this is some of the things that are connected to IP3 and calcium. Angina. I have acute angina. So angina is then, this isn't important. This is just to give you context for the next mechanism. But this is about the coronary arteries, which lead to the heart. So they feed your heart with oxygen. And when they get filled up with plaque, less oxygen can get to your heart, less blood and less oxygen, therefore. And you end up with pain in your chest, which could kill you eventually. You get hypoxic like that, you can end up getting arrhythmia. So you could go into a fibrillation and just die instantly from this. So this is not a good thing. Okay, so one of the things you can take for it is nitroglycerin, a temporary fix, basically. It's going to cause relaxation of veins, arteries too, but mainly veins. Here's how that works. You've got your smooth muscle cells in the wall of the vessel, and you've got endothelial cells on the inside of the vessel. Different neurohumoral factors like histamine, acetylcholine, a lot of different things can activate receptors on the endothelial cells and produce IP3. And I said this before, but everything we saw back here is involved in that one little arrow. So we're kind of shortcutting it because it's so complicated. But IP3 then goes to the ER, binds to IP3 receptor, releases calcium, binds to calmodulin, and that activates NO synthase. So NO synthase, or NOS, is going to take arginine and make nitric oxide, NO. NO being a small, wrong way. NO being a small molecule and nonpolar for the most part, will then diffuse into the smooth muscle cells and activate guanyl cyclase, which is going to act like adenyl cyclase, except with GTP, and it converts GTP into cyclic GMP. And then another protein kinase here gets activated, protein kinase G. Okay. And, we saw before that calcium causes contraction of the vessel here. Cyclic GMP causes relaxation of the vessel. So that's physiological antagonism.
Boy, if I could explain that to my patients.
It's a struggle.
Oh,
Okay, so here's another thing G proteins can do. You could have receptors that are linked through G proteins to channels. This is your potassium channel, really important in the heart. SA node, this is going to help control the rate of cardiac contraction. This alpha subunit can also activate some channels, usually like a calcium channel or something. But that's just another thing that G proteins can do, activating channels. And that affects the membrane potential and can activate the cell to do something. And this is this is the second type of hormone, which is your long-term response type hormone. So all of these receptor mechanisms will cause permanent changes in cells and take a long time to happen. Most of them are going to be growth factors. EGF, epidermal growth factor, transforming growth factor alpha, PDGF, platelet derived, TGF beta, fibroblast growth factor. These are cytokines, which is our JAK-STAT pathways. We didn't talk about hedgehog. You don't want to know about hedgehog, believe me. It's just another pathway, and it's another pathway. Okay. Here's how the tyrosine kinase receptor tyrosine kinase pathway works. Your receptor is a single peptide, and it's got a binding site for the hormone on the outside, a single alpha helix going through the plasma membrane, and then an enzyme, a tyrosine kinase here. And again, this is important because most of your kinases are serine threonine. This is a tyrosine kinase, which is going to only phosphorylate tyrosines. And then the tail has got a series of tyrosines in it as well. And these tyrosines, as I said before, it's not just the tyrosine, it's like the tyrosine plus 3 other amino acids next to it that will comprise an SH2 domain, okay? So when this gets phosphorylated, it's going to attract proteins with a binding domain to them. And each different consensus domain will attract a different protein. We talked about several that can come up to the membrane. SRC is an enzyme, phospholipase or phosphatidylinositol 3 kinase, GTPase activating protein, phospholipase C, SHIC, which is like GRAB2, phosphotyrosine phosphatase, negative inhibitor of receptor tyrosine kinases, GRAB2, JAK, STAT, SHIC, phospholipase C, STAT. So a lot of different things can be brought up to these membranes. And that's why, you know, what I tell you is like the brief discussion of what these receptors do. The reality is they're doing a ton of things all at once, and it's scary. It's scary sometimes. Sometimes I need to be held. Well, I'm getting punchy here. We better have an exam soon. Okay, so when the hormone binds to the receptors bind up to each other, and then they transphosphorylate, so you get all these SH2 binding domains here. That then attracts a protein like GRAB2. GRAB2 is an adapter protein. It binds to the receptor, and it also binds to an effector, SOS, son of sevenless. So that's one of the things that can be brought up here. So SOS then activates the small monomeric protein, G protein, RAS. So RAS is similar to RAC-RO CDC42. Those are from chapter 13, very similar in structure. So RAS is going to let go of its GDP when SOS is around and pick up GTP, and then becomes active. So this is G protein cycling, just like we saw with the large heterotrimeric G proteins. It will continue to shut itself off with the help of GAP, which is going to activate that G protein or that GTPase. So it continue to cycle as long as you've got SOS here to turn it on. RAS then will activate a kinase cascade, so 3 kinases in a row. Each of these steps is an amplification. So RAS activates RAF, RAF activates a bunch of MEK, MEK activates a bunch of MAP kinase. MAP kinase then goes into the nucleus and will phosphorylate Jun. Jun is half of a transcription factor called AP1. So when Jun is phosphorylated, it binds to Phos. Jun and Phos come together and make AP1. Now, AP1 is going to activate cell division, and we'll see that in the next chapter. So make sure you know this pathway, because in the next chapter, we're going to add a whole nother pathway onto it. And then you can say, you'll know everything from when the hormone hits the receptor to the cell going into S phase and starting to divide. So this is the first half of that pathway. So that's the receptor tyrosine kinases. And this is a little something I threw in there. Your dominant negative receptor, basically you make a construct of this. You get the DNA, you cut out the tyrosine kinase domain in the whole C terminal and express that in cells. And it's going to knock out the growth factor pathway in that cell. It's going to dominantly negatively affect all receptor activation in that cell. In this case, the FGF receptor. And you can see here, the way it's used is basically, you put that in an early embryo and you find out what happens when that hormone isn't present anymore. Knocks out that signaling pathway, and therefore you see what that signaling pathway was important for, because it's not there anymore. Okay, serine threonine receptor kinases. This is your TGF-beta-like pathway. This is a lot simpler than receptor tyrosine kinases, I think. You've got 2 different receptors, the type 1 receptor, the type 2 receptor. When the hormone is around, all 4 of those clump together, transphosphorylate, and then they phosphorylate the receptor SMAD. The receptor SMAD then binds to SMAD4, which is your cytosolic SMAD, And that becomes a transcription factor and goes into the nucleus and activates transcription. So much shorter pathway than the receptor tyrosine kinase. We're activating the receptor the same way, but then that receptor is going to phosphorylate the transcription factor directly, and it then goes on and produces changes in the cell. Here's the JAK-STAT pathway. Again, this is similar to the TGF-beta pathway. The receptor binds the hormone, and then that attracts the kinases. The receptor itself doesn't have a kinase with it. The kinases then transphosphorylate, and that superactivates the kinases, and they then phosphorylate the tail of the receptor, which then brings the stats up, STAT1 and STAT2. They've got an SH2 binding domain, so they bind to that SH2 domain, and then the kinases phosphorylate them. They let go, and because they've got both an SH2 domain and an SH2 binding domain, they dimerize, and then they are a transcription factor. They go in and change transcription. So that's your JAK-STAT pathway. PI3 kinase pathway then involves, again, our friend PIP2, but now we phosphorylate it a third time on the 3 position, and now it's PIP3. So this then attracts proteins with a pH domain. So the pH domain binds to that inositol trisphosphate, which brings the protein close to the membrane. So this is kind of a general receptor mechanism we see all over the place here, which is recruitment. These proteins were just in the cytoplasm, but once that IP3 is there, it gets recruited to the membrane, and now it's close to its substrates. So now it starts to become active. So, for example, PDK1 gets pulled up to the membrane, as does AKT. PDK1 is then close to AKT and phosphorylates it. So that's what these domains, these binding domains are all about. They're bringing things together. And P10 shuts this mechanism off. It'll take that phosphate off the 3 position and turn that back into PIP2. Okay, so P10 is the off switch here. Here's how the pathway goes, your receptor. And again, this is receptor tyrosine kinases. So same start as in the RTK mechanism. Hormone binds, receptors dimerize, transphosphorylate, and that brings up PI3K because it's got an SH2 domain specific for this SH2 binding domain here. Okay, PI3 kinase. It then phosphorylates PIP2 into PIP3, makes a bunch of that in the membrane, which then recruits all these proteins that have pH domains, and PDK1, PDK2 both phosphorylate AKT, and then AKT can phosphorylate other things. A lot of different targets for AKT. We'll see in the insulin receptor mechanism that it's got targets in that mechanism as well. Here, this is the main thing, though, that AKT is going to do. It's going to phosphorylate GSK3 beta. So GSK3 beta, normally in the absence of hormone, and this is all off, is going to be phosphorylating beta-catenin. And when it gets phosphorylated, it becomes a target for ubiquitination and degradation. So in the absence of hormone, this destruction complex is constantly breaking down beta-catenin. So beta-catenin levels stay low. When AKT gets activated, it inhibits GSK3 beta by phosphorylating it. Beta-catenin doesn't get phosphorylated. It builds up in the cell really quickly. It's a transcription factor. Goes into the nucleus and binds with TCF, LF, activates transcription. Here's a question. Why would the cell waste the energy to constantly make beta-catenin if it's only going to break it down? Why doesn't it make it when it needs it? Good question, Dr. Slish. So the idea is it's going to build up in the cell a lot faster if all you have to do is stop breaking it down, right? To cause a protein to come up, you have to activate your signal transduction pathway, turn on transcription, make the messenger RNA, transport it out of the nucleus, translate it, and then you've got the protein, right? All that takes time. In this mechanism, all that's going on constantly. You're constantly making messenger RNA, constantly making the protein, but it's constantly being broken down, and that keeps it low. When this pathway gets turned on, it quickly goes up. So instead of it taking, 45 minutes to come up, it'll come up in, a couple of minutes. So that's why this mechanism works this way. Here's your insulin receptor mechanism. I showed you my animation, which I think is better. But again, the receptors are just receptor tyrosine kinases. The only difference here with insulin is it's a heterodimer. Each half of the receptor is a heterodimer. Not that big a deal. Once it gets activated, it transphosphorylates, brings up this guy, IRS1, insulin receptor substrate 1, which then brings up our old friend PI3 kinase, making PIP3 again. And that activates AKT, PDK1, PDK2. And your AKT then activates or phosphorylates a protein on vesicles that have the glucose transporter in them. And that gets put up into the membrane. It also phosphorylates GSK3 beta again. And in this case, what's going to happen is you're going to stop phosphorylating glycogen synthase, which allows glycogen synthase to make glycogen. So now you've got glucose transporters in the membrane, you're bringing glucose in and you're turning it into glycogen. So glucose levels go high, insulin gets released, and then your mainly liver and muscle transport glucose in and make glycogen out of it. Blood glucose levels come back down. Oh, and here's TGF beta again with our destruction complex. So this is now being, this GSK3 beta is being active or inhibited in a different way through this mechanism. So we've got Wnt, LRP, frizzled and disheveled. So in the absence of Wnt, your destruction complex again is constantly breaking down beta-catenin. When Wnt is around, it binds to frizzled and LRP, and that brings disheveled up, and disheveled then attracts axin, and you make a big complex up here. And the whole point of this complex is to bring axin off, because once it comes off the destruction complex, GSK3 beta is inactive, and beta-catenin builds up, and the cell goes into cell division. Ooh, is that it? Yeah, I guess that's it. Okay. Thinking we had another pathway. I can give you 3 or 4 more pathways if you want, as long as we're here. I'm not busy. Okay, shifting gears slightly. From here, we're going to signaling in early development of xenopus. So we're only getting up to the point of gastrulation here. And kind of the point of this, and the reason I give this to you is give you an idea what these receptor tyrosine kinases and TGF beta-like kinases are for. What do they really do in the body? Really, really important here in early development. And if you're in my developmental biology class, you can skip this whole thing because this is a lot less than we talked about there. Okay, so we start out talking about, in general, about what gastrulation is and why. Some animals have got no axis of symmetry. Some have got one axis of symmetry, top and bottom, and they're radially symmetric around that axis. And some have got 3 axes of symmetry. You've got your anterior versus posterior, dorsal on top, ventral on bottom, and then right and left going into and out of the screen here. And there's my favorite quadrupedal animal. That's not my dog, but he looks like that. Okay, so how does this happen? How do you get your 3 axes? The egg starts out with 2 different poles to it, so you kind of have a radially symmetrical egg. The top of it is the animal pole, the bottom is the vegetal pole. And they're different right from the egg stage because you've got specific messenger RNAs attached to the cytoskeleton at the top and the bottom during the production of the egg. When sperm enters, that makes the decision where the ventral side is going to be. Sperm comes in the ventral side, the opposite side will be dorsal. After the first division, you've got 2 cells, both have dorsal determinants in them, so you can separate these cells and make 2 clones, two embryos that are exactly like each other. After the next cleavage, you can't do that anymore because you've got 2 cells here that have dorsal determinants and nothing ventral. And you've got two cells over here that have only ventral determinants and nothing dorsal. So if you separate these, none of those cells will become a whole frog. After the third division, the top is separated from the bottom, and now you've got two different tissues here. You've got ectoderm, which is determined by the fact by the messenger RNAs that are maternally placed there. So these are going to be ectoderm, and so their fate is ectodermal. And the bottom is endoderm, so they will be endodermal. Ectoderm makes skin and nerve. Endoderm makes gut. Watch that video, it's really cool. So division, 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 cells keep dividing, dividing, dividing. Eventually you end up in the blastocele, or the blastula. So this is the blastula stage with a fluid-filled cavity in the middle. The top, you can see, are much smaller cells. This is your ectoderm. Ectodermal cells divide faster, so they end up smaller. And then down here is your endoderm. Now, in order to get a complete embryo, we need a third tissue, which is our mesoderm. And that's going to happen by factors released from the endoderm to the ectoderm. That's going to change these ectodermal cells into mesoderm. So the mesodermal cells then form a ring around the middle of the embryo. And these mesodermal cells are going to become muscle, bone, blood. I think I told you this at the time, last time I talked about this, but I used to teach in that Kaplan course, and they used to call this mesoderm because it gives you the means of getting around. Mesoderm, that's how you remember it. So muscle, bone, blood, means. Okay. So now we've got the three different tissues we need, but we still only have one axis of symmetry. And because of that sperm entry at one point, the other point ends up being dorsal. So now we end up with having a ventral side and a dorsal side of this blastula. And this becomes apparent when the dorsal side forms a little lip. So you've got these bottle cells that will change their shape and cause this indentation. And then some of these mesodermal cells and some endodermal cells will come into the embryo and crawl across the roof of that blastocele. So here they are coming in and across the roof. And there's a gap here between endodermal cells that are ingressing. These are ingressing versus endodermal cells that are sitting there at the bottom of the blastocele. And so that makes a little gap between the sides, and that's the inside of your gut. That's the archenteron, the primitive gut. The blastocele will get pushed out, it'll disappear. And then once you're at this point, you've got three layers of tissue: ectoderm, mesoderm, endoderm. Okay, that's your three layers, and that's what you need for three axes. You've got over here anterior, over here posterior, this will be the butt, and then dorsal up here, and ventral down here where the stomach is. And while this gastrulation is going on, you also have a pibili going on the outside, which is these ectodermal cells dividing, 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 spreading out, coming all around the cell, or all around the embryo, until the whole outside of the embryo has ectoderm on it. Skin, you're gonna need skin all over you. Okay, so the next thing that happens, which we're not going to talk about in this class, but I want to throw it out here anyway, is once this comes all the way up this way, you then get neurulation that goes in the opposite direction, which makes your spinal cord. Okay, so here's that involution here, the leading edge here of the mesoderm coming in. These bottle cells are endodermal cells that face like that, and they shove that endoderm in. And now you've got mesoderm and endoderm, and they form a layer that then crawls along. So remember, as they're crawling, they're dividing, so you're getting more and more cells, and that's allowing it to go across. Okay, and there's your gastrula. After it's gastrulated, it's called a gastrula. Really good video. Okay, fate map. So important things to know, fate. So fate is, like, at any particular time, what is that cell going to become? That's its fate. And its fate can change based on hormonal interactions, but this is the fate map of a blastula if you just leave that thing alone and let it do what it wants to do. So you've got ectoderm on top. Some of that ectoderm will be neural, some will be skin. And then you've got mesoderm around the middle. You've got dorsal mesoderm. So this will be nodal cord, which will help form the spinal cord and other things like that. And then you've got middle mesoderm, your somites, and then ventral mesoderm, blood and blood vessels and stuff. And then your vegetal endoderm, which is just gut, like we could do without it. I love my gut. Induce. So induce then is when a hormone activates or a signal transduction pathway, which then activates transcription factors to come on. Those transcription factors then will activate a whole new raft of genes, and that will change the cell type. So now it's differentiated into something else. That's what induction is, and it causes differentiation, changing from one cell type to another by turning on different genes. And now you've got different proteins there. It's a different cell when it's got different proteins in it. Okay, mesoderm induction around that rim. What is this going to do for us? Three things. Number one, it's giving you that third cell type, right? You need muscle, bone, and blood as well as skin and gut, and that's where that's what mesoderm is going to do. Second thing it's going to do is gives you a dorsal ventral axis based on where that sperm came in. That's ventral. The other side ends up dorsal. Now you've got two axes, the first one plus that one. And the third thing is that mesoderm is going to cause gastrulation to happen, and that ends up setting up all three axes, posterior, anterior, posterior, dorsal, ventral, right, left. Okay, how do we know this happens? Early experiments before people had molecular tools, one thing they did to figure out where mesoderm was coming from was to take the animal cap off, dissect away any mesoderm. So now you've got only ectoderm. If you leave that ectoderm in tissue culture by itself, it will become epidermis. But you're going to take that cap, put it on top of the endoderm, and then you'll get mesoderm. You get an induction and mesoderm forms. So we're going to take this blastula, actually. This is all ectoderm up here. We're going to take that, make sure you don't bring any mesoderm with it. And we're going to take the endoderm without mesoderm, just this part here, put your ectoderm on top of it, and booyah, we get mesoderm from that ectoderm. So, and it's ventral mesoderm. So this tells you that there's something being released from the endodermal tissue that's causing ectodermal tissue to become mesoderm. And right here at the margin, we call this the margin between ectoderm and endoderm, that's where that factor has effect, and that's why you get mesoderm all around the middle there. Experiment 2, we're going to, so now at this 32 cell stage, if you look at these cells, and you can't really tell whether they're dorsal or ventral. It's still just a spherical ball of cells. But there are determinants there. There's already fate that's been determined. And you can tell that because if you take some of these cells out, put them in the opposite side of a different embryo, they gastrulate. So you end up with a whole separate axis. So this embryo here is going to gastrulate in two ways. Don't ask. So you end up with a pretty messed up embryo. So that's telling us then, and then here's the video on the Spemann organizer, and that gives you an even better axis. So point here is that something early on is causing this cell to be dorsal, while these are all ventral over here. And those dorsal determinants are going to be called an organizer because they're going to organize a new axis. They're going to cause gastrulation to happen. So our first organizer is the Newcoop center. And the Newcoop center then will cause the Spemann organizer to form in the mesoderm on top of it. So the Newcoop center is in the endoderm. It releases factors that cause the Spemann organizer to form. So either the Newcoop center or the Spemann organizer are organizers because the Newcoop makes the Spemann is the organizer. So either way, you get a second axis when you take those cells out and put them into other embryos. Okay, so now the molecular aspects of this. Why is this all happening? So as I said before, there are a lot of messenger RNAs that are maternally laid down. An important one is VEGT. This is a transcription factor, and this causes the endodermal cells to release XNRs and BMP4. These are serine threonine kinase, TGF-beta-like mechanisms. So they're activating SMADs in this ectoderm here, and that causes the ectoderm to become ventral mesoderm. Okay, so that's kind of the first thing that happens. But at the same time that that's going on, you've got this other factor that's going to give us our dorsal ventral. And that other factor is the cortex of the cell has got some proteins attached to it at the vegetal pole. And in the egg, at the egg state, they're all right at the bottom of the vegetal pole. When the sperm hits, that activates cortical rotation. The cortex rotates away from sperm entry, and that puts the dorsal determinants over here on the opposite side. So those dorsal determinants are disheveled, which, as we know, inhibits or binds to actin and inhibits GSK3 beta and GSK3 beta binding protein. Both these things are going to block GSK3 beta. Beta catenin comes up. So over here, we've got high beta catenin as well as VEGT. We've got both transcription factors there. And those two together are going to combine to cause these cells to become the Newcoop center. Okay, you've got beta catenin and VEGT, and so that causes the production of the transcription factor Siamois, and Siamois makes this the Newcoop center. So it is the Newcoop center because Siamois is going to cause these cells to release a lot more XNRs. So we've got XNR going all the way around, we've got BMP4 all the way around, but here we've got a high concentration of XNR. And concentration makes a big difference in early development. Low concentration, no concentration, nothing. Medium concentration, you get A. High concentration, you get B. Makes a big difference in development. So here we've got a high concentration of XNRs, and that causes the transcription factor goosecoid to come up in these mesodermal cells. That makes this the Spearman organizer. And that's where that's going to be the dorsal lip of your blastula, of your blastopore. Okay, that mesoderm with goosecoid in it is going to start that invagination or ingression. And the Spearman organizer is going to then pattern dorsal ventral. So we've been saying from way back here that this is ventral and this is dorsal, right? That's its fate. That's not what it is right now. And how you actually get dorsal versus ventral is these cells are fated to become the Spearman organizer. And once they do, the Spearman organizer is going to release cordon, noggin, follistatin, and XNR3. And these three things are going to block BMP4 activity. So you've got this point source of BMP blocking plus BMP being released everywhere, okay? So over here, BMP has got no activity because it's being totally blocked. Over here, there's no cordon and noggin because it's too far away, so there's high BMP4 activity. So BMP4 is basically the default. Ventral is the default. When the Spearman organizer gets formed, you get this gradient of BMP4 activity, and that makes this mesoderm dorsal, medial, and ventral. Dorsal, so if you look at from the top of the fish to the bottom, dorsal is that outer part, right, which has got your spinal cord and your spinal cord and vertebrae, ribs, okay, that's dorsal. You come down, you get into the body cavity, medial mesoderm, that's really ribs and muscle. You get to the bottom of it, that's where your heart is forming at the bottom, and that's the ventral. So that's how you get this dorsal to ventral mesoderm. But again, at this blastula stage, we still really only have one axis of, well, I guess we've got two now, one top and bottom, and then ventral dorsal. But where we really get all three is when this dorsal lip comes in and then pulls that mesoderm across the ectoderm. And then more things that tell us what we just talked. And then also those blockers will also block BMP4 up here, which will make this neuroectoderm instead of epidermal ectoderm. Okay, and that's all there is. That's all I'm asking you to know by Tuesday. Hopefully you've been studying all the way along. See, I always, you know, I tell people this every year, no one ever listens to me. After every class, you should be studying this stuff. When people say, oh my God, I've got three tests this week. Well, if you're studying all the way along, you're just reviewing, you know, you don't have to learn that material again. And I also suggest while you're studying, go back and look at exams 1 and 2, because that'll come up in the final as well. So refresh yourself. Otherwise, what always happens is, or what happens with a lot of people is, instead of reviewing all the way along, they wait till the week of finals and it's like, I can't study all this. And they give up and they do, you know, as well as they can off the top of their head. So keep reviewing. And that's what I have for you today. Oh, don't forget the game is up there. The game is due before the exam starts. So make sure you're doing that game. And questions, comments? Okay, I'm gonna let you go. I miss you guys when you're gone all weekend. We should get together on the weekend and have a Zoom too. All right, later guys.